Thank you. Hi, everyone. As Sir mentioned, I'm giving this talk on behalf of the authors. I'm not one of the authors because they couldn't make it, unfortunately, to the conference. So there will be some omissions and errors, but you can blame me for those, not the authors. And this talk will be about creating a data release mechanism that allows us to control the trade-off between privacy and utility in the context of genomic data sharing. So we all know that increasing amounts of genomic data are being collected and shared for various applications, including personalized medicine, direct-to-consumer genomic testing, which is popularized by companies like 23andMe, as we heard this morning from the keynote speaker. And these data are shared in different contexts. So for example, in the public setting, there are online platforms like OpenSNP, where users can publicly share their private genotypes for recreational or research purposes. And in the clinical setting, a patient's genome may be used to inform clinical decision-making or support medical research. And due to the private nature of genomic data, there are various data protection mechanisms that have been put in place. So examples include technical measures like secure storage and transmission of data, authentication for access control, data use agreements, which allows to limit the scope of how these data are used after being shared. There are also laws and policies like HIPAA in the US, GDPR in Europe, which more broadly regulates different types of personal information. One common strategy to protect individual privacy in these settings is to limit the type of information that is shared. So for instance, there are these public databases where we openly share only aggregate level genomic information, such as summary statistics, or simply presence or absence of different mutations in the cohort. However, even these limited forms of data sharing are known to be vulnerable to what's called membership inference attacks, where statistical tests are used to determine whether a given person's genome is included in the data set or not. And the concern here is that there could be other metadata associated with the individuals that are considered sensitive. So if a data set includes only individuals with a specific medical condition or disease, then knowing that person is in the data set could be considered a serious breach of privacy. So over the years, many approaches have been developed to address these risks. One example is this approach that aims to release only a subset of variants that are uncorrelated as a way to prevent correlation-based data reconstruction attempts. Presence or absence queries, these themselves are considered to be a safer way of sharing these data compared to, say, sharing the actual counts, mutation counts in different cohorts. Optimization and game theoretic approaches have also been explored, which try to incorporate additional factors that influence the feasibility of these attacks. For example, the costs associated with carrying out these attacks in practice. And lastly, randomization-based approaches, including those based on differential privacy techniques, and also perturbing rare alleles in the data set, which are considered more sensitive, have also been explored. So the main limitation with these approaches is that most of these techniques, perhaps with the exception of differential privacy, don't really provide a principled way of controlling the trade-off between privacy and utility. And here, by utility, what I'm referring to is the usefulness of the data that is being shared, which could be affected if we're applying some sort of perturbation to the data for privacy. And this often leads to a large utility loss for these methods, which is a key barrier. The other limitation is that all these approaches either consider either data suppression or noise addition as possible defense mechanisms, but never both, even though using both of them at the same time could lead to better privacy and utility trade-off. So let's look at the model of this work in a little more detail. We'll be considering only biallelic variants for simplicity. So for each haplotype variant, we either have a reference allele or an alternate allele, so it's encoded as 0 or 1. We'll then consider two different data sharing scenarios. So the first one is Genomic Beacon Services, which was introduced in 2015 by Global Alliance for Genomics and Health, or GA4GH. So these are services that answer presence or absence queries that I briefly mentioned earlier. So for each query, we're looking at whether there's any individual in the data set that has a particular genotype at a specific position in the genome. The other setting that the authors consider is referred to as summary statistics here. And specifically, the authors are referring to a service that returns allele frequencies of different variants computed in the cohort. Now, in terms of the attack model here, remember that we're considering potential attackers who have access to the genome of a private individual, and their goal is to figure out whether that person is in the data set or not. And the authors consider a prominent type of attacks that are based on likelihood ratio tests, or LRT. You can think of it as, think of this as a way of probabilistically reasoning about membership of different individuals in the data set using knowledge about population level frequencies of different genotypes, different variants. And in particular, for these tests, we'll be computing a score or a statistic that captures the ratio of likelihood between the alternative hypothesis, where the individual, the target individual, is in the dataset, versus the null hypothesis, where the individual is not in the dataset. And this ratio is compared to a certain threshold to return the decision about the membership inference. And there are two different settings for this part. So the first setting is using a fixed threshold. So this is a constant threshold that's applied across all the individuals in the data set. And the idea here is that this could be optimized on a separate data source to maintain a reasonable level of false positive rate. The second, more challenging attack setting is using adaptive threshold, where the threshold value is different for each target individual. And these are obtained by using a secondary data set where we estimate the empirical null distribution. These are the main contributions of this work. First, the authors introduce a novel optimization framework, which helps us tune the privacy utility trade-off in the context of sharing these summary level genomic data. And second, they introduce algorithms that combine both data suppression and noise addition to achieve this. And this defends against LRT attacks. Just a couple things to clarify here. So suppression here means withholding completely withholding information about a subset of variants that are chosen for both beacon queries and allele frequencies. On the other hand, noise addition means slightly different things depending on the setting. So for beacons, the noise refers to randomly flipping the yes or no response depending on the variants that are being queried. For allele frequencies, the noise will be a real value number that's sampled from a Laplacian distribution. And this is inspired by the standard Laplace mechanism from the differential privacy literature, if you're familiar with the field. Another contribution of this work is that this is optimizing this problem is done by these scalable algorithms that the authors developed, which improves both utility and privacy and scales to a realistic setting with a large number of variants, with more than 1 million. So the problem that the authors introduce is called Summary Statistics Privacy Problem, or SSPP. Just to go over a few notations here, M here defines the set of variants that are masked or suppressed. Delta is a vector of noise values that are being added to the remaining variants. Z is defined to be the set of individuals whose privacy is protected once the proposed mechanism is applied, depending on the values of M and delta. And this is the objective function being optimized, which has 3 terms. So the one with delta captures utility loss due to the addition of noise. The second one with M captures the utility loss due to masking of data. And then the third one with Z is the privacy loss term, which is set to be the negative of the number of individuals that are protected. And again, this is a function of M and delta, which controls the behavior of the mechanism. And the idea is to jointly optimize these 3 terms with respect to M and delta. There are a couple of additional parameters here that control the relative importance between these different terms. So alpha here controls the cost of noise addition compared to masking, and W controls the cost of privacy loss compared to utility loss. And the insight behind these parameters is that we can now vary these parameters to map out the trade-off for different settings to inform our decisions about the most promising strategy. Okay, so given this problem formulation, the authors introduce a couple of greedy algorithms for optimizing this objective. I'll briefly go over the process here. So in the case of Beacon, for each genetic variant, the algorithm will be computing a score that measures the marginal impact that a variant has when we're masking or flipping it on the success of the LRT attack. And so all the variants are sorted according to this, the score, additionally incorporating the weighting terms based on the parameters alpha and W that I mentioned earlier. And once we have this ranked list, we'll greedily walk down the list to apply perturbation to the most impactful variants. And during this process, the list is continuously updated as we apply one perturbation at a time. And we stop this once we reach a reasonable privacy level. And the same approach is applied to the summary statistics setting with alloy frequencies, except that here we're alternating between masking variants and adding Laplacian noise to the allele frequencies that are returned. So here are the experimental results. We first consider the fixed threshold setting, where again we use the same threshold for all the individuals. This is showing the Beacon query problem here. There are two plots. The left one considers baselines that only flip the variants. The right one shows baselines only masking the variants. Author's approach is shown in blue, labeled SPG. There are 4 other baseline methods here that are all based on published works. So DP is differential privacy, which is a standard way of adding noise to the data to achieve privacy, which gives rigorous but often highly restrictive privacy guarantees. RF is random flipping approach, which focuses on hiding rare variants in the data set. Both SF and MIG are also greedy algorithms that are close to the author's approach, but using a different scoring scheme. And the key difference of the distinguishing factor of the author's method is that they jointly consider both flipping and masking, and they also consider alternative privacy settings where we're not just going for like the full privacy protection, which these 2 methods do. So overall, we see that the blue line sits on top of everything else, so the author's method performs quite well. And it's worth noting that the MIG method, it's a little hard to see here on the slide, but it's being shown as a single marker in the highlighted regions here, behaves similarly to SPG, but again, this doesn't allow this fine-grained trade-off between privacy and utility. And here we're looking at the results for the allele frequency setting, where we consider a different set of baselines. We have DP, again here, there's a pure masking-based strategy, and then the linkage one is referring to the one that I mentioned earlier, where we filter out correlated variants. And here, an interesting parameter here is alpha, which again controls the cost between flipping and, adding noise and masking a SNP. So when alpha is set to 0.9, which is shown on the left, flipping, adding noise, is highly costly. And this is why you see that the masking approach is highly effective here. And SPG follows closely behind. But once we reduce the value of this parameter, SPG now outperforms the masking-based solution, and it overlaps with the DP solution. And the key benefit of SPG is that it gives a unified framework for interpolating between these two different approaches. Now, we're moving on to the adaptive threshold setting, which again is a more challenging attack scenario. So these plots are showing the same thing as what I showed you before, but now in the context of this new attack model. This is also for beacons. And you can see that SPG outperforms other methods in both compared to both flipping-based baselines and masking baselines. And we have a similar observation here where the MIG achieves similar performance as SPG, but it doesn't have that trade-off property. And on the right side, there's something interesting going on here where all the baselines have 0 privacy. And what this means is that in this challenging attack scenario, only masking the variants is not enough to gain any privacy protection. And so SPG is able to combine masking with flipping to obtain a version of this mechanism that gives full privacy guarantee, full privacy protection with minimal loss in utility. And lastly, this is looking at adaptive threshold setting with allele frequency queries. The authors noted here that a low value of alpha here makes SPG reduce to a simple flipping-based solution. Sorry, the one that's only based on addition of noise. So here they're only looking at alpha values of 0.75 and 0.9 in this example. In both cases, we see that the SPG outperforms the other baseline methods as well in this setting. And one interesting thing to note here is that both linkage and DP approaches in these cases do not approach full privacy protection within the range of utility that's being plotted in these figures. So in conclusion, this work introduces a novel optimization framework that allows us to tune the trade-off between privacy and utility. This is done in the context of sharing genomic summary data. And this is achieved by introducing these scalable algorithms that combine masking and noise addition and improves both utility and privacy over the existing methods. The authors would like to acknowledge funding support from the NIH and NSF, as shown on the slide. Since I'm not involved in this work, I'll take questions offline, but you're welcome to send your questions to the first author at the email that's shown on the slide. And thank you for your attention.